Our global food system is under, well, incredible pressure right now, isn't it?
It really is.
Climate change, pushing crops to their limits, a growing population needing food, and this strange situation where we have so much, yet so much deficiency too. It feels like we need more than just small tweaks.
Absolutely. The traditional agricultural toolkit, based on methods decades old, it just isn't equipped for the precision or the speed or the adaptability we need now. It's
Yeah.
a moment that really demands transformation, not just, adaptation.
Okay, let's unpack this then. Today, we're diving deep into some truly revolutionary science.
Yeah.
CRISPR and synthetic biology. These aren't just buzzwords anymore, are they?
Not at all.
They're a powerful suite of tools that let us design, edit, and, well, accelerate the evolution of food right down at the molecular level.
Yeah.
Our mission for this deep dive, to explore exactly what we can do and what problems we can actually solve with this incredible technology. We'll look at how we can engineer the future of food making, everyday vegetables, calorie-rich, building crops that are climate-ready, virus-secure. It's pretty amazing stuff.
Before we jump into the solutions, though, let's just frame the challenge. It's huge. By 2050, the planet needs to feed nearly 10 billion people.
10 billion.
Wow. And at the same time, we're seeing usable farmland actually shrinking, extreme weather getting more common, and traditional crops just failing under these new environmental stresses.
The need is critical.
Undeniably. There's a critical need for new solutions, yeah.
Okay, so this is where it gets really impactful, right? We're talking about CRISPR-Cas systems.
Yeah.
And they offer this incredible precision,
Unprecedented precision.
allowing targeted edits to a plant's own DNA without necessarily introducing foreign genes. It's like a molecular spell checker for the plant's code.
That's a good way to put it. It lets you pinpoint and rewrite specific instructions.
So we can, silence genes that limit how much a plant produces?
Or enhance genes that improve nutrition, yes. Or build plants that are optimized for specific environments, specific resistance profiles.
And you pair this with other tools, like RNA interference, RNAi.
Right, RNAi for silencing specific genes. And there's also CRISPR-Cas13, which targets RNA, making it great for fighting viruses.
Okay.
Add in speed breeding protocols, and you've got this technological convergence. It's really reshaping agriculture into something data-driven and rapidly responsive.
Let's start with a big one then, something concrete we can solve. Engineering food to be more calorie-rich.
A major challenge.
Yeah, when you think about the, over 2 billion people suffering from malnutrition globally, it's not just about getting enough food, it's getting enough nutritious food, energy-dense food.
Exactly. And historically, vegetables, they're great for vitamins, for fiber.
And low on calories.
Relatively low, yes.
So how can gene editing help solve that?
Well, intriguingly, tools like CRISPR-Cas9 and RNAi can fundamentally rewire this. Scientists can now directly modify the plant's internal metabolic pathways.
The cellular machinery.
Exactly. The machinery responsible for how it makes and stores starch, sugar, protein, lipids, fats.
So what does this actually mean for, the food on our plates?
It means we can turn traditionally low-energy crops into, bioengineered sources of caloric resilience. It's a real solution.
Can you give an example?
Sure. There was a study, Liu and colleagues, they used CRISPR-Cas9 to switch off 2 specific genes in rice. Now, these genes normally kind of suppress the plant's ability to use energy efficiently, especially during drought. The result, rice plants that yielded significantly more grain, and not just under drought conditions, but also with normal irrigation.
And that wasn't from adding chemicals or anything external?
No, not at all. It was about unlocking the plant's own inherent potential, tweaking its existing systems.
That's powerful. So we're not just adding calories, we're optimizing how the plant itself allocates and stores energy.
Precisely. And we can also directly enhance, say, starch production pathways.
How?
Well, in potatoes, for instance, researchers successfully edited the gene responsible for a certain type of starch, amylose. They created these waxy potato variants.
Black soup potatoes.
Yeah, it means they store more amylopectin, a more rapidly digestible starch, so a denser caloric profile per potato.
Right.
And similarly, Ghosh and others noted modifications to specific genes in maize can increase the synthesis of this energy-rich amylopectin, better caloric availability per gram.
Which is hugely valuable in humanitarian context, I imagine.
Extremely valuable, yes.
It's not just about carbs, right?
Yeah.
You mentioned fats and proteins. Can we really get more of those into vegetables?
Indeed. According to Mala and Yang, you can use combinations of RNAi and CRISPR. You could say silence internal compounds that inhibit fat production.
Turn off the brakes on fat storage.
Sort of, yes. Or you could activate regulators that boost lipid synthesis. researchers have already managed to increase oil content in research crops like Arabidopsis and Camelina. And early trials with leafy greens are looking promising. Imagine like an oil-rich lettuce or protein-packed kale.
That sounds like science fiction.
It's scientifically conceivable and potentially deployable in communities where animal protein access is limited. It solves a real dietary gap.
That is a game changer, truly. And can these changes be combined? Can you stack these traits?
That's a key consideration, yes. Precision trait stacking.
Meaning,
Meaning you simultaneously modify multiple pathways. So maybe you tweak how plants transport sugar, how efficiently they photosynthesize, and how they allocate nutrients.
so you get a plant that does everything better.
Essentially, yeah, crops that not only grow faster, but also channel more of their energy into the edible parts, the high-energy tissues. This could totally redefine caloric yield per unit of water or light or even just time.
You can target where this happens.
Exactly.
Yeah.
Using genetic promoters, you can tell the plant, activate these high-energy genes only in the parts we eat, like the tubers or the leaves, minimizes waste.
So it's a shift from just growing things to actively designing them.
It's a transition from passive cultivation to active metabolic engineering, yeah.
Yeah.
Building a smarter plant from the inside out, solving the energy density problem at its source.
Okay, let's shift gears a bit. Agriculture has always felt, well, slow, governed by seasons.
Painfully slow sometimes.
Developing a new crop variety, maybe with just one new trait, used to take, what, 10 to 20 years?
Easily, sometimes longer.
And in today's climate, with things changing so fast, that timeline just isn't viable anymore, is it?
Not at all. We need solutions much faster.
So this is where another aspect comes in: speed.
Right. This is where the convergence of CRISPR editing, streamlined genetic engineering systems, and these speed breeding protocols really transforms things. How quickly we can modify and multiply food.
Okay, tell me about these engineering systems first.
Well, systems like Golden Gate or Gateway Assembly, they let researchers build complex multi-gene constructs really rapidly. It's almost like snapping together molecular Lego bricks. Very efficient.
So you can edit not just one gene, but multiple genes at the same time.
Yes, exactly. That's called multiplex editing.
Multiplex editing.
Yeah. Ghosh and colleagues, for example, describe using CRISPR to simultaneously edit 3 key genes in rice. One controls grain number, another grain size, and a third affects how dense the seed head is.
3 at once.
3 at once. And this triple edit produced plants with significantly higher total grain yield, didn't mess up the plant height or how long it took to grow either.
And that would have taken how long traditionally?
A decade, maybe more,
Yeah.
through conventional crossbreeding. They achieved it in a single generation. That's solving the time problem.
Incredible. And the speed part isn't just the editing itself, right? You mentioned speed breeding.
Precisely. Speed breeding is about optimizing the growth environment to accelerate the plant's life cycle.
How do you do that?
You use controlled chambers. You can give them extended daylight, up to 22 hours of light per day.
Wow.
Optimize temperatures, nutrients. Plants like wheat, barley, chickpea, they can reach reproductive maturity in basically half the usual time.
So you combine the fast editing with fast growing.
Exactly. Put the CRISPR-edited plants into a speed breeding system, and researchers can get through maybe 6 generations in a single year, instead of the usual 2 or 3 outdoors.
6 generations a year. That acceleration means huge real-world impact for agricultural resilience, doesn't it?
Absolutely. If you connect this to the bigger picture,
Yeah.
think about a world where droughts, floods, new pests, they strike with very little warning.
Which is increasingly our world.
It is. The ability to quickly develop and deploy new resistant varieties, it's absolutely crucial. What once took a decade for, say, a pathogen-resistant variety might now be done edited, tested, scaled in less than 2 years.
That agility is vital.
Vital, not just for big industrial farms, but crucially for smallholder farmers. They're often the ones facing the immediate brunt of climate disruption. Faster solutions help them adapt.
And you mentioned open source platforms earlier. How does that fit with speed breeding?
Yeah, that's important too. The fact that speed breeding works well with open source synthetic biology platforms means this innovation isn't necessarily locked up by large agribusinesses.
So others can use it.
Universities, NGOs, maybe even citizen scientists, they can participate in co-creating crops adapted to their specific region. It has the potential to be a tool for equity, for democratizing this power, not just for efficiency.
Okay, so we've talked about boosting calories and speeding up development, but there's another battle, right? This quieter war against viruses.
A huge, often underestimated problem, yes.
Plant RNA viruses cause billions of dollars in losses every year. They cripple staple crops, cassava, tomato, maize, rice.
Devastating impacts on food security, and traditional methods, well, they're just not keeping up with the viruses.
So what can these new tools do about viruses? How can they solve this?
This is where the next frontier of gene editing comes in, CRISPR-Cas13 and also RNA interference. These tools essentially defend the crop from the inside by targeting the viral RNA directly.
Okay, you mentioned Cas13 targets RNA, unlike Cas9, which edits DNA. How does that help with viruses?
Well, many major plant viruses are RNA viruses. Cas13 is like a precision-guided missile for RNA. It binds to and cuts single-stranded RNA in a sequence-specific way.
So it intercepts the virus's instructions.
Exactly. It shreds the instructions floating around inside the plant cell before the virus can replicate effectively.
So it's like giving the plant its own built-in programmable immune system.
That's a perfect analogy. As Chaudhary and colleagues describe, you can engineer guide RNAs to recognize stable, conserved parts of the viral sequence. This means the plant can resist even viruses that mutate rapidly.
That's clever.
And it has this other feature, collateral cleavage.
You sound dramatic.
It kind of is. Once Cas13 is activated by finding its target viral RNA, it doesn't just cut that one molecule, it starts cutting other nearby single-stranded RNAs too.
Like a molecular shredder going wild.
A bit, yes. It basically disrupts the whole viral replication cycle very effectively. Now, there were initial concerns about it potentially damaging the plant's own RNA.
Right, that sounds risky.
But researchers have engineered newer Cas13 variants with much reduced collateral damage. They're safer, more specific now.
Okay. And how does RNA interference, RNA AI, fit into this viral defense picture?
RNA uses the plant's own natural machinery to silence foreign genes, including viral genes. Molla and Yang explain it's been successfully used to give resistance against many plant viruses, and crucially, this resistance can be inherited by the next generation.
So you can use both, CRISPR and RNAi.
Yes, it's a powerful combination. RNAi could act as like the first line of defense, maybe intercepting viruses as they enter the cell. Then CRISPR-Cas13 acts as the heavy artillery, targeting and destroying any active viral RNA that gets through.
A layered defense system.
Exactly, creating truly immune-engineered plants, fortified from within.
The implications for global food security seem enormous here.
They really are. I mean, the key question is how much food can we save? Think about cassava mosaic virus in Africa.
A huge problem.
Affects over 40 million tons of cassava production every year. Or tomato leaf curl virus can wipe out entire fields.
It's devastating for farmers.
Absolutely. A single CRISPR-edited line, maybe with this Cas13 immunity built in, it could save millions of tons of food, stabilize local markets, and reduce the need for chemical pesticides. That's a triple win.
And looking further ahead.
Well, more futuristically, this kind of precision immunity opens the door to maybe real-time viral monitoring within the plant, plants that sense and respond proactively.
Plants become their own security systems.
In a way, yes. Solving the vulnerability problem before it even starts.
Okay, so we've covered these incredible possibilities. More calories, faster breeding, virus immunity, amazing potential for solving major food challenges. But this power brings serious ethical questions, ecological concerns too, right?
Absolutely. It's like any powerful technology. It has immense promise, but also significant perils. You have to look at both sides.
What are the immediate concerns?
Well, one is intellectual property. Who owns these tools, these edited traits? CRISPR-Cas systems are already battlegrounds for patent disputes.
Right.
If traits for, say, disease resistance or enhanced calories become proprietary, locked behind expensive licenses,
Then smallholder farmers, developing nations.
it could be shut out, limited access. It could actually reinforce the inequalities we already see in the global food system.
That's a major worry. And what about the environmental side and the ecological impact?
That's another big area. Using CRISPR widely raises questions. What happens if a superproductive, climate-resilient engineered plant escapes cultivation and outcompetes native wild plants?
Reduces biodiversity.
Potentially. Or leads to unforeseen gene flow, the engineered genes getting into wild relatives. We don't always know the long-term consequences.
And the viruses could fight back?
There's always that potential, yes.
Yeah.
Viruses evolve. They could overcome the engineered resistance, maybe even faster than we can edit new defenses. And Chaudhary and others also caution about that Cas13 collateral cleavage. It needs to be really tightly controlled to avoid accidentally harming the plant itself long-term.
So the big question becomes, how do we guide this technology? What direction should it take?
This leads us right to ask, Should this biotechnology primarily serve the market, or should it serve the planet and its people?
Because the first wave of GMOs,
Often prioritize traits like herbicide resistance or longer shelf life, things that benefited producers or distributors, maybe more than consumers or the environment.
but CRISPR and RNAi offer a chance to change that.
There's an opportunity here, yes. A chance to reorient biotech towards broader human and ecological needs. Things like increasing food sovereignty for communities, enabling local adaptation to climate change, maybe even using engineered plants to help restore degraded ecosystems.
But that depends on who's driving the research.
Exactly. It depends entirely on who funds it, who directs it, and who ultimately gets to deploy these tools. The solving part depends on what problems we choose to solve.
Are there any positive signs on the ethical front? Moves towards making this more accessible, more responsible?
There are some encouraging signs, yes. Significantly, we're seeing the rise of open source biology movements. Public CRISPR libraries, like the ones Ghosh mentioned for speed breeding platforms, these are actively trying to democratize access.
Spreading the knowledge and the tools.
Right. And collaborative international programs are emerging too, offering training, bioinformatics tools to scientists in low and middle income countries, aiming to share the benefits more broadly.
That's good to hear.
It is. But even with progress towards inclusion, we have to remain, well, humble. Genes don't exist in a vacuum.
They're part of complex systems.
Incredibly complex. Biological systems, cultural landscapes, planetary cycles. A modification that seems beneficial in the lab, like increasing calories, might inadvertently reduce a plant's natural medicinal compounds or change its role in the local ecosystem in ways we didn't predict.
Unintended consequences.
Exactly. As Mala and Yang emphasize, even when the editing tools are precise, the real-world consequences might be emergent, not easily predictable. Biotechnology gives us incredible power, like a paintbrush.
But we need wisdom to guide the hand holding it.
Precisely. Human vision and wisdom must guide its application.
Let's try and paint a picture of that future then. Imagine a world where famine isn't just seasonal, where maybe skyscrapers grow food vertically. And every seed is tailored not just to its climate, but maybe to the specific nutritional needs of the people eating it. That's the kind of future these technologies hint at, isn't it?
Right.
A future where we genuinely use science to solve hunger and food insecurity on a global scale.
Absolutely. In that maybe not so distant future, CRISPR, RNAi, speed breeding, they don't just enhance agriculture. They could transform it into an intentional adaptive ecosystem.
So high-calorie crops wouldn't just be grains anymore. We could have
You could have leafy greens like kale or amaranth, bioengineered to store energy-dense starches without losing their vitamins. Tomatoes might express genes to convert sugars into beneficial lipids, a natural energy source where cooking oil is scarce.
protein-rich vegetables.
Becoming staples where meat is hard to come by, yeah. Crops optimized for nutrition, climate resilience, and maybe even cultural relevance. And in drought-prone regions, edited plants could use, say, root-deep expression of water-sensing genes to thrive where others just wither.
And these traits wouldn't be fixed.
No, that's the key. They'd be selected, iterated, improved season by season using those speed breeding cycles. Crops could adapt almost as fast as the climate shifts.
Wow.
You could have entire plant libraries maintained in digital genome banks, open source, maybe community managed, allowing for rapid deployment of the right seeds for the right conditions.
And the viruses. Would they still be wiping out harvests?
Maybe not. Crops could possess these programmable immune systems we talked about, RNA-based biosensors detecting pathogens, activating CRISPR-Cas13 on demand.
So the plant defends itself instantly.
Yeah. In the rice field, the maize plot, the plant responds with molecular precision, neutralizes the threat before you even see symptoms, and if a new viral strain pops up...
They update the software.
Essentially, yes. Scientists could update the plant's immune script with a new guide RNA, distributed almost like a software patch. Constant vigilance, molecularly encoded.
And this isn't just for huge farms out in the country. It sounds like it could reshape fooding cities, too.
Absolutely. Cities could become living agricultural systems, rooftops, balconies, indoor vertical farms, all growing custom-edited crops matched precisely to urban dietary needs and constraints.
Food becomes truly local, adaptive.
Minimal waste. And meanwhile, maybe in remote rural areas, you could have community biotech labs, empowering farmers to clone, test, even edit their own seeds.
Breaking free from dependence on large seed companies.
Breaking free from monocultural, top-down structures, fostering real, local food sovereignty.
So the vision isn't about replacing nature, is it? It sounds more like working with its fundamental building blocks in a smarter way.
That's the hope. If we connect this to the bigger picture, the whole ecosystem could become more participatory. Gene-edited crops could potentially work with nature, maybe enhancing pollinator health, rebuilding soil through better nitrogen fixation, regenerating degraded land with specialized pioneer species.
So the old split between natural and artificial starts to blur.
It could fade, yeah. Replaced by a new paradigm, perhaps. Thoughtful co-creation with biology itself.
We really are standing at the threshold of a new agricultural epoch, aren't we? One not just dictated by passive inheritance, but by the conscious design of life.
It's a profound shift.
We're not just working around nature's constraints anymore, we're collaborating with its code. The crops of tomorrow won't just hopefully survive climate change, they might anticipate it.
They won't just endure disease, they could intercept it.
They won't provide calories just by accident, but by deliberate intention. It really comes back to what we can do and solve.
But the future of food isn't just a technological question. It never is. It's deeply moral, ecological, political. We have to decide, does this incredible power to edit life become a tool for equity? Or does it become a weapon of monopoly?
Are we designing food to reflect values like resilience, sustainability, justice?
Or are we just scaling up the old logic of extraction, just with more efficient genes this time? These are the critical questions.
The science is here, or rapidly arriving. The question is, what do we collectively do with it? In a world that's increasingly shaped by algorithms, by viruses, by AI, food remains this fundamental connection.
To the earth and to each other, sacred, really.
Yeah, and what we choose to grow, how we grow it, it reflects who we are. With tools like CRISPR in our hands, we're authors of a new agricultural story.
We have choices to make.
We can choose to write one version, calorie-rich vegetables accessible to all, virus-proof fields, democratized innovation. Or we could write a different story. Consolidated control, ecological blindness, maybe even digital hunger.
This future genome, it's still being sequenced. We're writing it now, but the outline is becoming visible. Food that can heal, adapt, and feed everyone, hopefully without asking the planet to keep paying the price.
The next harvest won't just be of crops, will it? It feels like it needs to be a harvest of wisdom, of careful design, and of shared responsibility.
That sums it up well.
So much to think about. What stands out to you about this incredible new chapter in agriculture? We really hope this deep dive has given you plenty to mull over.